Hi, you guys. Welcome to my mini lecture on DNA. So when we first start thinking about DNA, it seems like the extraction these days is so easy, right? And you see it on shows and they get DNA and they're able to extract it and they're able to find out who done it. But it's actually not that easy. And this ability that we have today, though, of using DNA has been really important when you think of the law. So there have been 312 post-conviction DNA exonerations in the U.S. Think of that. 18 of those 302 people exonerated through DNA served time on death row. I mean, this is a big out. The average length of time served by exonerees is about 13 years, over 13 years. And the total number of years served is approximately 4,000 if you were to add up all those years of all these individuals. So it's a fantastic tool that we have today that's been helping out in the court system. So if we were to look at a karyotype to take a better look at it, this is a karyotype. And what that means is these are all of our chromosomes for a human chromosomes. And we have 23 pairs of them. We have 23 pairs because we have one from one parent, one from mom, and one from dad. One from mom, one from dad. And they are arranged in these discrete little clumps, and we have 23 of these discrete little clumps inside our cells. So if you were to add and glue each one of those together, you would have your entire length of the DNA for a human. It just turns out that it's in these discrete little pieces. And each one of these little pieces then is going to carry a gene for something. So it could be for whether you have attached or detached earlobe, could be on chromosome 3. On chromosome 7, you could have something like second toe longer than first, and so on and so forth, all the different traits on these 23 pairs of chromosomes. When we look at a chromosome, why are we seeing them like that when we usually think of DNA as this big ladder? And that is because during reproduction, while the cell is dividing, that DNA is going to scrunch up and it's going to ball up into these little packets, and those little packets are chromosomes. You don't see them do that when the cell isn't in the middle of cell division. So it only does it when it's dividing because it's easier to scrunch them up and then move them places than have this tangle, this ball of yarn of chromosomes of DNA that's hard to divide up and put in one cell and in the other when you're having cell division, when cell division happens. Okay, so that's how usually we see them. We see them as this X, and this is already a duplicated chromosome. So if you follow my arrow, this here is a chromosome. And then when it duplicates, you see it in double. So this causes a lot of problems because if I were to show you just one of these, it's a chromosome. If I show you two of them, it's still one chromosome, but duplicated, instead of saying that it's two chromosomes. Do you understand? That can be really confusing. But we only see them when they're duplicated, so we're going to see them as this X. When we look at the vocab for what a chromosome looks like, each leg of these we're going to call a chromatid. So one chromatid, another chromatid, making up really one duplicated chromosome. And then if we were to pull out, like a piece of yarn, pull this out and stretch out that bunched up DNA, you would eventually see it like the double helix with its base pairs, which is A and T and C and G, and we'll see a little bit more about that. Okay, so this is what the structure looks like when you see it three-dimensionally. And these long strands, then think of them as a chromosome that is scrunched up together. So to discover this shape was a big deal, and it wasn't easy. And it was done by a group of scientists. Many of them worked on this, and they were all desperate to try and find out what the structure was. One of the important people was Rosalind Franklin because she was taking X-ray crystallography images where you take photographs And she came up with this sort of X image. It's kind of like a chandelier. If you see the light hitting a chandelier and you see the shadow that it forms, you could potentially find out what the shape of that chandelier was given the shadow. And that's how she was trying to figure it out. She died before the discovery was actually presented to the world of science. So she wasn't included in the Nobel Prize of Watson and Crick that we hear so much about. And she was really quite crucial, but didn't make it into that prize. So when we look at the shape and we want to look at these nucleotides, remember we talked about those. DNA is composed of nucleotides, and what a nucleotide is a phosphate, a sugar, and a nitrogenous base, meaning it's a molecule with nitrogen on it. This is a nitrogenous base. So always you're going to have these nucleotides. Only what's going to vary is which nitrogenous base you have. There are 4 only nitrogenous bases: adenine, thymine, guanine, and cytosine. So they're all going to differ a little bit here in what they look like. I'll show you what they look like. And they're going to be able to bond to each other and form this long chain. So in this long chain here, you're seeing G bonding with C, A bonding with T, C bonding with G, etc., etc., and forming this very long chain. The fascinating thing about this is that everything is made up of DNA. It's a tree, it's you, it's a rabbit, it's a fungus. It's made up of A, C, G, and T, which is sort of very similar. It's remarkable. They had a questionnaire that went out to many Americans saying, do you think that all foods that have DNA should be labeled? And a bunch of people said, oh yeah, we want to label DNA. That's kind of strange because we're all made up of DNA. Everything has DNA. All your food has DNA. It's not something you have to label. Okay, so what we want to look at is, how do these connect? How do these pairs connect? And there's my nucleotide in a box again. And what I want you to notice first is the hydrogen bonds. So hydrogen bonds connect complementary DNA strands, as I'm showing you. Notice that when it's a C and a G, it has 3 bonds. 1, 2, 3. C and G, or G and C, 3 bonds. When you get to A and T, it's only 2 bonds. And then here again we have 3, and then 3, and etc. So on this next slide, then I broke it down so that you just have one set. We have T and A, which is thymine and adenine, and C and G, just cytosine and guanine. Notice that because of the electrons that they have to share, if it's A and T, it's going to have 2 bonds that bond to these 2 molecules. And if it's C and G, it's going to have 3. So if I were to remove these labels, I take them away and I say to you, okay, tell me. I mean, you don't have to decipher everything that's going on here. But if I would ask you, is this a bond that has to do with thymine and adenine, or is it one that has to do with guanine and cytosine? And you'd be able to tell me without really recognizing every detail of the molecule because of the number of bonds. Okay, these bonds, I want to go back. Remember, it is a nucleotide with a G, a nucleotide with a C, and these 2 are bonding together to form the ladder, to form the middle of the ladder, the rungs of the ladder. Okay, what else can we notice about this? Other than that, we want to know that adenine has a double ring and thymine has a single ring. And the same goes for guanine. Double ring, cytosine, single ring. So now, if I didn't tell you, if I, again, removed these labels and said to you, label this for me, what would you do? You'd start out by saying, well, these 2, it's got to be T and A or A and T because it's got 2 bonds. What else do you know about it? Well, if you remembered that adenine has 2 rings and thymine has 1, then you'd be able to identify this is adenine and that's thymine. Even though the double bond here and the oxygen, you know nothing about that. You just know that the rung of the ladder here has to be A and D, and you just read DNA. So if I give you a long sequence, you'd be able to read it. For this one then, if guanine has 2 rings and cytosine has 1, then I would know which is which. Great. So now, if I give you a strand, you would be able to read it. All right. You have more practice with the other videos, not done by me, but external videos, and those are helpful too. So don't forget to look at those also. Bye.